It's great to be here. I'm Jonathan Sweedler. I'm a chemist, and you might ask, why is a chemist in CABBI? And hopefully some of that will be clear. And so we've heard a lot today about some of the goals to make particular chemicals, make the plants into manufacturing. And that obviously takes chemical analysis. Are we making the right or the wrong compound? This has been a really fun period here because I've been introduced to some measurement challenges I never thought I would have to do. And one of them is that many people in this room are amazingly good at automating and basically coming up with samples where they say, hi, I have 10,000 samples to measure, or 100,000, because the chemical engineers can, like Huiman, can automate a process and do error-prone PCR. My research group and others that do this crazy technique called mass spectrometry tend to have done it serially, where it takes 10 or 20 minutes a sample. I always thought that was fast until you say you have 100,000 samples. And then what happens is all the undergraduates run. And so what I've really want to talk to you today about is some of the approaches that we've done to make our measurements worse, but really, really high throughput. And one of the reasons I put this slide up is Huiman keeps on talking about the wonderful automation, and it is just amazing. But I like this picture of the road because if you have an 8-lane highway between Chicago and San Francisco and you make it 16 lanes, but you still forget to build the bridge over the river, it really doesn't go any faster. And if the slow point is the measurements, then we really have to address this. And so that's what today's 101 talk is. I'm sorry, it's analytical and sort of, okay, it uses mass spectrometry and lots of flavors. I can't ask how many people know what mass spec is? See, oh, there is a few hands, that's good. And so anyway, I'm trying to keep this less technical and we'll see how we go. So one sort of general slides, we need the chemical information to make the decision. Most people are aware of the techniques of gas chromatography mass spec and liquid chromatography mass spec. It's wonderful. It is the gold standard in the sense it gives you a lot of chemical information, but it's serial. So what I'm going today is to talk about a few techniques that we have available, and we're making it available not just to one theme, but now making measurements throughout CABBI that make this a little bit faster. And they're just a slide or two on each of them, and then I guess I will say my team and I are here for the whole meeting, and just talk to us if you have any questions. The two first techniques that we really developed in the first few years are, okay, there's a lot of different names for them, but they're just high throughput approaches. One of them, if you want to think of it, is a little bit like, I'd say Star Wars, but it's maybe more like Star Trek in the sense that you have a bunch of colonies, you have a bunch of spots, you actually figure out the locations and you shine a laser at it. I call it like Star Trek because we always hit our target. So if you have 10,000 to 100,000 colonies, then we have 10 to 100,000 samples. We don't do a separation. We lose all the advantages of liquid chromatography or gas chromatography, but it's now a second to sample. And so a lot of this is how to get your sample. So when I talk about the sample, by the way, I have to admit, this has nothing to do with how long it takes you to prepare the sample. If that takes you 5 minutes or it takes you 5 years, that's not on me. So when I say a second per colony, and then of course, then there's a lot of informatics, and we're still working with a lot of groups on how to deal with that. And the second one is, that approach is fast. It gives up some quantitation if you need that. There's this liquid extraction surface analysis. I apologize for all the things. And this is can provide absolute quantitation if you need that. It works with volatile samples, but it's a little bit slower. It's about a minute a sample. So just a slide on both, but it's just sort of showing you how this works. I won't use the pointers, you don't really need it. But the idea then is you have an array. It could be randomly organized spots. It can be a microarray. If you image it and figure out where the locations of the samples are, then our mass spectrometer will use a laser or an ion beam and sample each one. And it's quite fast. And the second approach, just a little bit more information, does provide an absolute amount, and they have different flavors, and so they work a little bit better if you're working with, because of the way the mass spectrometers work, a little bit better if you're working with isomers or if you need low molecular weight or volatile materials. Both of these are available, and we're using these for more and more samples. These are sort of somewhat unique. If you talk to most mass spectrometrists in your facility, they'll say these aren't as good as, for example, something like gas chromatography or liquid chromatography in terms of precision and quantitation and complexity. Absolutely true, but if you also tell them I need large numbers of samples, you'll also probably not make as much progress. I thought I would spend the next few minutes talking about other flavors of high-speed measurement. This one I'm calling Where is Waldo, because if you think about a plant, you might not want to know just where your molecule is, or what the molecule is, what particular alcohol or fatty acid you're making, but you want to know where it is. And that's something called mass spectrometry imaging. This is something that a couple labs in the CABBI are very, very good at. We've been doing this for quite a while, and so is Josh's group. And I just wanted to mention this. This is showing a brain slice because I had it, but it could be a leaf. We're working with Andrew Leakey on this, for example, and you can see an array of dots. In this particular case, we just basically make an image just like you would with your phone, except maybe it's 100 by 100 or by 1,000 by 1,000 array. That would be 1,000,000 pixels. It's low resolution, but every pixel, instead of being 3 colors, has 1,000 or so, in this particular case, lipid information. If the dots are hit with a laser, we can get a few micron resolution. If it's an ion beam, we can go down to nanometers. And so the spatial resolution depends. Nobody would ever image an entire plant at the nanometer scale because you would have many, many terabytes of data even if you could. And so there's also other information. It's not a new technique. I just put this slide on the write-up because it's one of the first examples. Franz Hillenkamp, the inventor of this flavor of mass spectrometry, actually showed some example in the 70s, but it's gotten a lot better. Let me just show you one example. It's the first one we did on a ginkgo leaf. This is not one of the CABBI crops, but we actually imaged miscanthus in 2010 for a DOE effort. And so we've been working on this for quite a while. And again, you can get a lot of information on where molecules are. Is it high throughput? Per image, it's not. But if you're actually interested in looking at 50 spots on a leaf, if you're looking at a lot of different spots, in a microbial ecosystem, then for region of interest, it can be quite high throughput. This is, again, something that our instruments can provide, and if you're interested in knowing localization, let me know. Again, it's not, if you don't need localization, just because you can, it's not something you would want to be doing. One last area, and that's single object mass spectrometry. It sort of fits what I was talking about before, and this is good for metabolomics. I sort of liked this because in 2023, I guess that's last year, Nature editors picked it as one of the technologies to watch. I kind of always wonder about showing this slide because now it's 2024. I guess it's no longer a technology to watch, but I'll still talk about it. And this sort of puts everything into perspective in terms of state of the art. You can scatter 10,000 cells. You can put a leaf on there and just look at the guard cells. This is just showing a microscope slide with some teaching targets with about 10,000 cells, in this case in green. I had it for a brain. It works just the same for anything else. It doesn't matter. And then we hit each individual object wherever they fell, wherever for example, the leaf put the guard cells, and just hit those objects. Again, we never miss an object within a few microns. What we can do then is if you scattered cells, you get individual cells, and you get a lipid profile in every individual cell or every individual location. And this works surprisingly well, and again, not counting sample preparation. It's about a second or so per sample. Now again, that's the acquisition. If I hand you a terabyte of information on thousands or hundreds of thousands of cells, it can take a month or more to go through the data set. So it's not like, this is the, think of an observatory where you acquire over a night and then you spend the next 6 months going through the data. So I do have to emphasize that it does take a while to figure out what exactly we're trying to do with the data. And it's one of the reasons there's a lot of back and forth in the beginning. And then just recently, if you want to say what is state of the art, this we published with Van Lam and others, and this was actually doing imaging, it was doing single cells, it was doing serial slices. It would have been many, many months of time to acquire this data set, and so Van Lam, as an informatics person, came up with some compressed sensing approaches and others to speed this up about 100-fold. We came up with other ways, and you can actually then truly acquire a lot of information and put a serial stack together and get information on, in this case, really designed for lipids, fatty acids, and others. And again, partly because it was, Nature methods, it was really emphasizing some of the aspects that were for a brain. But every one of these do work in plants. And it gives you this ability to actually compare what's in the individual cells, what's extracellular, and how to put this together. In terms of an application here, it would really be fun to look at the root microbial ecosystem to see where metabolites are. Let me give you one different example, and this is some work with Hudson and others, and that's some work we're doing with single cell, big cells, protoplasts, where we're trying to, this allows us to see that the protoplasts have the particular metabolic profiles before we grow them into full plants. And so this is some work where we're working with the Hudson Lab and the Zhao Lab and others, where we're automating the gene cloning, we're the transformation, and then we're doing this, the single cell lipidomic measurements being MALDI. We've compared this with the gold standard and made sure that it works. This allows us to get a lot of information from individual cells very early in development. We're now adapting this to make sure, it's one of the editors on this submitted wanted to see something besides lipids, that it works for sugars and others. The one thing I'll say about mass spectrometry is that part of it does work, and so there won't be a problem with that. And so we're then going to be able to expand this. But if you have a need for working at a very small level, then that's also a possibility. And so I've sort of really quickly, I noticed, we're already over on the session, talked about high throughput for MALDI, which allows, very high throughput, and then LESA for surface analysis, and then how to take this for imaging, as well as single objects or single cells, single guard cells, for example, if that's what you need, or even surveys across a broad range of chemical and spatial information. And again, I'm always amazed at some of the clever ideas that people have every time I come to this meeting and ask questions that really drive some of these future studies. And so I and my team are here for the entire time. Ask us questions. We'll figure out if your ideas are feasible or science fiction for another year or so. And with that, I thank you very much for your attention.